All right, chapter 9 notes. Your vocab words, there's aerobic respiration, it's with oxygen, anaerobic respiration without oxygen, active transport, diffusion, surface area to volume ratio, ram ventilation, pump ventilation, osmosis, water potential, stenohaline, euryhaline, osmoconformer, and osmoregulation. Just some quick bio review that we actually did in class together. Respiration is not breathing, it's the oxidation of glucose and then thus the release of energy in the form of ATP. Every single cell has to do this. Our more active cells have more mitochondria, so they could do this more efficiently. Aerobic respiration is able to carry out that process with the presence of oxygen, and it actually yields about 36 or 38 ATP molecules. In anaerobic respiration, the cell can only go through glycolysis, or the mitochondria can only facilitate glycolysis, which is the breakdown of glucose, but it's not complete. You only yield out 2 ATP molecules. So your body can produce energy whenever it's really low on oxygen, but it's not going to yield as much. Respiration will create ATP. ATP is adenine, a ribose sugar base, and then you have 3 phosphate groups. When one of the energies are used, or when an ATP molecule is used, you will lose the phosphate bond at the end, and then you're left with adenosine diphosphate. 2 phosphates are left. Okay, active transport. So active transport is going to be the movement of molecules against a concentration gradient, going from where you have a low amount to where it's already high, so going up the gradient. An example of this is going to be a fish muscle cell. It's a really high concentration of glucose, and yet it's going to want even more glucose. This is very similar in our own body. So glucose is going to be pumped into a cell that already has a large concentration of it. So it's going from a low amount to where it's already high. Carbon dioxide is released in respiration, and it must be removed because it's going to create lactic acid in their cells. So organisms need to be working at an optimum temperature for their enzymes to work properly and to be able to expel their carbon dioxide. And so you've seen a graph like this in chapter 8 when you looked at photosynthesis. Excuse me. I haven't had coffee today. All day. It's bad for everyone. Gaseous exchange, so diffusion across a semi-permeable membrane. Diffusion is just a random movement of particles from a high concentration to a low concentration until they spread out because they want to be in equilibrium. I have a couple examples here. Putting food coloring in water and seeing it disperse is an example of diffusion. Okay, so what's going to affect diffusion rates, or how fast things will disperse randomly? Temperature. As temperature rises, the kinetic energy of your particles will also rise, and they're going to move faster. They're going to bombard and hit into other molecules and cause them to also disperse a little bit faster. The concentration gradient, so the difference between 2 concentration amounts or 2 different amounts. If the concentration gradient is really large, diffusion is going to happen a lot faster. Respiration will use up a lot of oxygen, so oxygen concentrations are kept really low outside of your cell. That allows, when the oxygen is used up, it's going to flood right back in really fast because that concentration gradient's pretty steep. Marine organisms also use the same boundary inside and outside of their body so that we can move things really fast. Diffusion distance. So for diffusion to be really fast, the distance has to be really close. So the further the distance is, the further things are, longer it's going to take to diffuse. I'm sorry for yawning. Gills have really thin muscle, really thin, walls in between their, the lamellae, the little thin, extending pieces. Those are really, really thin, so gases can diffuse in really fast. They don't have a far way to go. It's not very thick. It's a small little area that it can diffuse through. And then the surface area. So organs like gills or our lungs have very large surface areas so that we can take in a lot of anything. And for this case, it's going to be oxygen gas. And that large surface area is going to help. Oh, my gosh. The large surface area is going to help diffusion happen faster. Okay. Water is the gas exchange medium, meaning that gas exchange is going to be happening through the water. Our gas exchange happens in the atmosphere. but marine organisms are able to extract gas out of the water. Their organs need to be really, really efficient because oxygen levels in water are about 40 times less than what it is in air. So they need to be even more efficient than our lungs at extracting oxygen from the water. Oh my gosh, this is going to be really hard. Oxygen is dependent on temperature and dependent on salinity, like whenever it's in the ocean. So you've seen this relationship before in chapter 7. The colder the water is, the better it is at holding on to dissolved gases, like dissolved oxygen. And the more salinity that you have, or the more dissolved particles, it's almost like the less space that the gas has to be inside of the water because it's taken up by so many other particles. So that gas concentration will also go down whenever it's more saline. Here's a fun little graph. It has two y-axes. You have dissolved oxygen on your y, and then on the other y, you have salinity. No, you don't. No, you don't. Sorry, they're both dissolved oxygen, and the salinity is based on the different colors. But what we're looking at here is no matter what, as temperature increases, no matter the salinity, your dissolved oxygen decreases. And the greater the salinity, the less dissolved oxygen there is. And that's a chapter 7 relationship. Water is more dense and it's more viscous moving through the body, and that's going to require a lot more effort, which is why in that pump ventilation, they have to close their mouth and lift up the buccal cavity to force it out because it is more viscous and it does require more effort. We can't just have it trickling through. Organs like gills need to have an inlet and an outlet aperture, and so they do. They have the mouth, and then they have out by the operculum. Aperture means opening. Our lungs only need one, our mouth, because air is way less dense and less viscous. It'll just fill it. Okay, so this is just showing surface area to volume ratio. Okay, and looking at this, you want to have a greater surface area to volume ratio if you're an organism that is going to be doing like diffusion through their own tissues. This is also why it's important we have such large and empty, I don't want to say necessarily empty, but we don't have huge volume inside of our lungs. It is pretty empty so that we can take in gas. The gills have very exposed. They're not thick. They're not deep. They're very exposed. They have a very high surface area to volume ratio. So you can see here like your smallest cube. And if you had a little nucleus inside there and something had to diffuse through and get into the nucleus, to have a higher surface area to volume ratio, like a 6:1 versus a 1.5:1 with this larger object, has way more larger volume. If gas needed to diffuse to get to the nuclear area, it's going to have to go a lot. Like it's going to have to go a lot further, and then it's not going to reach its destination as efficiently. So having a high surface area to volume ratio is good. You want to have a lot of surface area and really expose the tissues. I was trying to make this a little bit smaller. I can't. I can put it here. Okay. So this is an index that gives a relative measure of both the surface area and the volume, and they're compared together. If there's a really high surface area, that's lots of exposure, there's going to be really fast diffusion. Very high volume, very slow diffusion because it's thick. It's going to take time for it to reach on the inside. Larger organisms are going to have a really large oxygen demand. So body parts that are protruding and sticking through, like tentacles or the lamellae of your gills, that's really good for helping increase their surface area. Single-celled organisms do not need lungs and they don't need gills because they're already so small, gases can easily diffuse across their membrane. And as an organism's size increases, as the organism gets larger, their surface area to volume ratio goes down. Their surface area is not as great compared to their volume. As they're growing, they're going to get thicker and get wider. And yeah, they're going to have the same surface area, but the volume is going to increase. Therefore, that ratio is going to go down. And it's not going to be as efficient for just diffusion gas exchange. You're going to probably need a larger respiratory organ to facilitate that. Okay, fish gas exchange. So they have protein called oxyhemoglobin, or sometimes it's even myoglobin. It's really similar to hemoglobin. It's also red, and it will, because it's red, it's because it has iron. The iron will attract oxygen, and so that will help carry oxygen to the cells. Oh my gosh, this is awful. I'm sorry. Okay. So the path of blood in a bony fish. Let's see what the next slide entails real quick. Okay. Blood's going to pass through the organs and into the capillaries, which are your tiny, tiny, smaller vessels that are near your cells, so they will allow gas to diffuse through. Cells that are low on oxygen obviously are going to get the oxygen because of diffusion, going from a high concentration to low concentration. And the blood is going to gain the carbon dioxide because the blood should be deficient in carbon dioxide, while the cells that had just used oxygen and oxidized glucose are going to have a surplus of carbon dioxide. And so that's going to go into the bloodstream because it is a low concentration of it. The blood is returned to the heart in veins, and because veins go back towards the heart. Blood is then pumped out of the heart and the arteries towards the gills, and the blood passes through the really, really thin capillaries in the gills. It's going to release the CO2 that it just gained, take on the oxygen that just got put through, as they either did pump ventilation or ram ventilation. And again, then the blood's going to leave the gills, goes toward the arteries to deliver more oxygen and continue to remove carbon dioxide. Look at that. You can literally see like particles moving and blood moving. You can see them going in opposite directions. So where you have veins versus where you have arteries. How exciting. Okay, and they're not touching each other. They're moving next to each other. It's pretty awesome. Flick's law. So this is going to be for gaseous organs, and it's going to talk about the rate of diffusion. So the rate of diffusion, or particles going from a high concentration to a low concentration, is figured by taking the surface area, multiply it by your concentration gradient, and over the distance that particle is going to have to diffuse. So this law will allow us to predict gaseous exchange organs if they have a large surface area, if they have really steep concentration gradients, like a big difference between oxygen and carbon dioxide, and a short diffusion distance. How coral polyps do gas exchange. So they all do it through diffusion, through their tentacles, which is why they'll bring themselves out and allow themselves to kind of be within the water and get their carbon dioxide removed and oxygen taken in. It's all through diffusion purposes. They do not need gills. They don't have respiratory organs. It's all because of particles moving from a high to low concentration. The movement. If the water is pretty stagnant, if there's maybe not a strong current going through, they can create their own current by moving themselves back and forth. And you can see here they're so thin, so thin. So you can have obviously water coming in, they can take in water and carbon dioxide, so they can do photosynthesis. Once within the zooxanthellae, that oxygen that's created can then get diffused into the coral, the organism, so that it can use it to do cellular respiration. Fish gills. So bony fish like tuna and grouper can breathe by taking in water through their mouth and passing it over the gills and then forcing it out. Tuna is going to be doing ram ventilation for the most part, although it can also do pump ventilation. Grouper is our major pumper. Big groupers. Anyway, they have that gill covering, that gill plate called the operculum, and that's what's going to help open and close to allow gas to flow, or water that's full of gas to flow through and get in contact with their gills. Both sides of the head have gill arches, which is a bony structure that supports the gills. The gills contain tiny, tiny filaments called lamellae, and those have really high surface area, or they allow it to, it's very exposed. They're not thick, so the volume is small, but they're very exposed. The surface area is high. And these tiny capillaries inside the lamellae are going to extract the oxygen and move it through the blood. Okay, so all this is just zooming in even more and even more. You can see water can pass right through. Oxygen gets absorbed. There's your diffusion. Okay, we've watched this a bunch of times. Countercurrent exchange. So between the water and the gills, and again, this is really just another diffusion. Blood is going to flow through the capillaries. Here's your capillaries, all these small little webbings. Blood flows through the capillaries in the gills, inside the gills, in an opposite direction to the water. That way, there can be a really steep concentration gradient established and maintained, and then oxygen can move into the gills. So notice the way oxygen is moving through the gills and the way water is going to come through. Down here, we have a really low amount of oxygen. And this is when our water is going to start to, let me start over here. When the water, we have a really high amount of oxygen there. And as we need that steep concentration gradient. So as this starts to come through, it has such a low amount. That's going to be really steep compared to the really high amount that's in water. And this is going to be oxygen that's in gills that's already used. It's going to diffuse through because they're going opposite each other. One is going to have a lot, the other has a little. It's going to increase the diffusion. Okay, you did a much better job than I did. So looking at their diffusion gradient, counter current, it's a current that's almost going like counterintuitive, like backwards of what you would expect. And watch that video again. If you didn't, you just skipped over it. You're missing it. But you want to have, a steep concentration gradient. That's one of the things that will help diffusion happen fast. And you don't want to be able to put yourself in equilibrium. Marine mammals do this, they have lungs. So this isn't with their gas exchange. They do it with heat. It's called countercurrent heat exchange. And they essentially have their arteries and veins going next to each other, but that's going to be a way that they maintain body temperature. It's going to be a way that they can cool themselves down and then heat themselves back up. Okay, so mainly, like you said, your most fresh oxygen is going to, your most fresh oxygen is going to be, obviously in the front of the lungs, but the blood coming in that needs to take in oxygen is going to start at the end of your gills first, and then hit the most oxygen-rich environment whenever our blood is at its lowest, after it's already diffused the gases through. Okay, 2 types of ventilation. Ram ventilation is done when you're constantly swimming, keeping your mouth open. Water is just flowing through your mouth, passing over your gills because of the force of it. Ram ventilation, you got to stay swimming. So you're constantly swimming. So you're not actively pumping with your mouth and your operculum and buccal cavity. So you're saving energy there. However, this can only happen while they're swimming. Pumped ventilation is using the muscle contraction of the mouth to pump water through the gills. You're actively pumping, so actively using energy. But over here, it doesn't matter. In REM ventilation, you're constantly swimming. This can occur while swimming and at rest, so that's a benefit. Ram ventilation, an example of this is going to be tuna. So they're swimming so fast, it's going to force water through their mouth and out their, rubbing against their gills and then out their operculum. They don't need any kind of muscle contraction necessary in their respiratory organs to do this. It's all because they're swimming in a forward motion. Pros to this, energy is saved because they're not having to do muscle contractions. Cons, they're using a lot of energy because they have to be swimming. The gill structures are really delicate. They're really tiny. They could be damaged from a lot of water passing through, really high water pressure. That water has a lot of particulates in it, could be damaging to it. So to counteract this, tuna have like fused gill arches. And you can see all their tiny little mammalian surface area is super thin to allow water to pass through and extract oxygen. Okay, pumped ventilation. Do yourself a favor. Yeah. We saw this one in class. There you go. Open the mouth, operculum's closed. Close the mouth, operculum opens. Okay, actually, we already saw that in class. Okay, so the majority of fish can actively pump water over their gills, even while they're at rest, which is helpful. I just sneezed. Okay. They're going to use muscle contractions in their buccal cavity, like the cheek cavity of their mouth. That's going to require energy, but at least they don't have to swim. Pros, they can be stationary in doing this. Cons, it's energy costly because they're moving muscles to do this. The oxygen demand is going to increase. Your pumping rate is going to increase. So if the water gets warmer, you're going to have to pump more. Around times of an algal bloom, you're going to have to pump more because there's going to be a lot less oxygen in the water. But water always flows from an area of high pressure to low pressure. So inside the buccal cavity, when they open it, the water pressure is going to be really low. So water is going to flow in. Okay, we memorized this. If you didn't memorize this, then you probably missed 3 days of class last week. You have to memorize this. So in pumped ventilation, the mouth is open, and the operculum's closed. The pressure in the mouth is low, the volume in the mouth is high. This is going to force water to move in. When the mouth is actually open, it's the buccal cavity that drops. And then once the water's in the mouth, the buccal cavity lifts, the mouth closes. This increases the pressure in that small area, and it decreases the volume, forcing water to go out through the lungs. Whoops. I'm sure that was a really cute face I just made. Out through the gills, and it forces the operculum open. We've seen this one too. Okay, regulation of salinity. Osmosis is just the movement of water from an area of high water potential to low water potential, or an area where it's more fresh to where it's less fresh. Definition of water potential, it's the measure of potential energy that water will have in a solution for it to move from one place to another. The more dissolved particles you have in a solution, the lower water potential that you have. Different types of solutions, and this is all bio review. And this is always you're comparing the cell to the solution it's in. So when the solution, or the organism, so when the solution is hypertonic, the organism or us or our cells are in a solution that has more dissolved particles than us. That's what the prefix hyper means. In an isotonic solution, we or our cells or the fish are in a solution that is an equal concentration to them. And in a hypotonic solution, this can cause water to flow in, but it's because we are in a solution that has less dissolved particles than us. It's more fresh, and so it has a higher water potential. Living in different salinities. Living in the ocean is very hypertonic, so organisms are going to have to be able to adapt to having a high water gradient and a solute gradient. So some organisms can be stenohaline, which can only tolerate a smaller, narrow range of salinity changes. Other organisms can be euryhaline and live in deltas or tide pools, estuaries, lagoons, and they can tolerate a wider range. An example of this is salmon. I said in class today, go for salmon, pick salmon. Yeah, I'm excited. I'm almost done. Almost done. Okay, osmoconformer is an organism that is going to conform to its environment. We did this one in class today. Let me go grab it really quick. Refresh you. Okay, so this is what I'm talking about. Osmoconformers. I don't want to say they're typically stenohaline because they really are typically euryhaline. And euryhaline is an organism that can tolerate a wide range. So, many times they're invertebrates. They have that outer shell. Let's just... Oh, gosh. They can... What? One second. Okay. They can tolerate a wider range of salinity because they have adaptations to do so. That's what makes them euryhaline. They can live in a wide range. They can deal with salinity changes, and they have adaptations to allow them to do that. So your freshwater example of this, there's a lot of freshwater fish that do this. Like haddock is an example. Mussels are euryhaline. They can... Yeah, see, I even had it right there. Okay. They can survive many estuarine habitats. They can survive tide pools, things like that. Because when the salinity changes, they can close their shell up. They can also do some active transport, and they can match the solute concentration to their surroundings as to what's inside of them, so that there's no net gain or net outflow, inflow of water. Whatever comes in is equal to what goes out, and they reach an isotonic solution. Oh, goodness. All right, an osmoregulator. They're going to maintain their own internal salinity, and these are organisms that can regulate their internal salt and ion concentration. So let me see if my stylus will work on here. If it will, I'll annotate this. If it won't, hit up or down to change the size. Okay. Okay. I'm really close to this. I'm just going to quickly do what we did in class. Move this on the quickly. So looking at our water potential, inside the fish is going to have a higher water potential. Oh, gross. Let me get this together for a second. Hit up, down to change size, left, right to change color. Oh! Alright. I just evolved. Okay. Here we go. We're going to do high water potential. Low water outside of it. So it's going to be losing its water constantly. Also losing it out of the gills. One of its adaptations is to drink water. Drinks a lot of water. When it urinates, it's going to urinate a very small amount of water out. And then let's look at our solute concentration. I'll do that one red. When it urinates, well, we'll start with the solute concentration before I go to urination. It is more salty, higher salt. This is really sloppy. Sorry. Lower salt on the inside of the fish because it's a saltwater environment. So water is going to flood, salts are going to flood into the fish. When it drinks water, it's going to inadvertently take in some salts. And so how does it counteract that? It's going to do active transport, and it's going to take the already high amount of, the low amount of salt that it has and put it out to an area that's already high. So going from a low concentration to a high concentration, that's going to allow them to get rid of a lot of their salts. Okay, and that's your active transport. And then they're going to have very diluted urine, a small amount, but very diluted. Now in our freshwater fish, here we have low water potential, high water potential because it's fresh. So naturally, water is going to flood into this fish, into its gills. What it's going to need, though, is to get rid of all the excess water and keep its salt. So then when we look at the salt concentration, it has a higher salt quantity, salt concentration than what's on the outside of it. Salt diffuses, so it goes from a high to a low concentration. So they're losing salt, but gaining water. So when this fish eats, it's going to take on a lot of salt, or that's where it's going to get a lot of it from. It's going to do active transport of salt into its gills. So it's taking something from a low area, so it's taking salt from where it's already low and moving it to where it's already high. And that's going to be through active transport, moving it into the gills. When it urinates, it's going to urinate a lot of water. Lots of water, and just a little bit of salt is going to come out with that because it's going to want to maintain the salt. Okay, so what we did in class was a lot more cleaner. This is sloppy. What are you going to do about it? Oh, yeah, there we go. Okay. Nice. Oh, that just cleaned it. Okay, cool. That's awesome. All right, I'm learning. I don't need to go through this again. Okay, where am I at with my slides? Okay, really just 2 more. Cool. So how marine fish will adapt to salinity differences. And we like literally drew this in. You drew in the adaptations a couple times. So water surrounding the marine fish is going to be hypertonic, so it's going to have a greater amount of dissolved particles and lower water potential. Water is going to diffuse out of the fish because it wants to try and hit that equilibrium gradient. Salt's going to diffuse into the body. And just refer back to this, where our arrows are going. Constant water loss is going to lead to cell damage and death, and they're going to like shrink up and be dehydrated and have no water to do transport or send oxygen anywhere in their blood, so then they die. Instead, they're going to constantly be drinking water, even though it's saline. They have adaptations to go around that. They're going to be constantly drinking water to replace water loss through osmosis. Sodium and chloride ions are actively secreted by the gills in active transport. Little gill filaments on the outside, like outside the lamellae, they have protein pumps on the outsides that will actively push out salts into an already highly saline environment. It does require energy. Magnesium and sulfate ions are removed from their urine, and then the water that's now fresh is reabsorbed back into the body for the fish to use. And so when they urinate, it's very concentrated, but it is a low volume. And then freshwater fish and euryhaline fish that can tolerate a lower salinity if possible. So they're going to be moving substances across their membrane against the concentration gradient. It's going to be their active transport. That's going to be used a lot. So proteins on the cell membrane are going to pump ions out against the concentration gradient. So this is going to require ATP. Hold on. This is marine fish. Freshwater fish. They're not pumping ions out. Let's change that. They're going to pump them in because they're losing. They're losing. So they're pumping it in. This is my fresh water. They're pumping it in. Okay. Freshwater fish, they have the opposite problem of marine fish, and I kept pointing that out to you guys in class. Look at how your arrows are different. One's losing water, one's gaining salts, one's gaining salts and losing water. I'm just done. I'm just done. I just said the same thing twice. Okay, it's 5 o'clock. All right, so the water, if it's hypotonic, this is going to be a freshwater solution compared to the dissolved particles inside of the fish. Again, it's going to have the opposite problem of the marine fish. Our freshwater fish is going to be taking on a lot of water and losing its own salts to the environment. So what do? They're going to not go out of their way to drink a ton of water. They're going to get a lot of their salts from the food that they eat, and they're not going to go out of their way to drink a lot. Their gills are going to pump sodium and chlorine ions into their bodies. So it's active transport because it's going from an area where there's low solutes, which is the fluid it's in, into an area that's already a little bit higher. So it's going to go against your concentration gradient. And then whenever they urinate, they produce large amounts of very diluted urine because they're getting large amounts of water just flooding their body all the time. Oh, that was kind of a sloppy end there, parrot. This is cute. You can actually see the sun corals moving around and eating and getting some oxygen. And all right, cool. Done deal. Awesome. This is the shortest lecture you guys have had. See you tomorrow.